there's pretty much no intervention that works the same in both sexes. Either they work much better in one sex than the other, and that suggests that there's a fundamental difference in the way we age.
From the USC Leonard Davis School of Gerontology, this is Lessons in Lifespan Health, a podcast about the science and scientists improving how we live and age. I'm Professor George Shannon, Kevin Shue Chair in Gerontology. On today's episode, how Professor Bérénice Benayoun is advancing what we know about how cells age and can possibly be returned to a more youthful state by focusing on areas that have historically been overlooked. When Professor Benayoun joined the USC Leonard Davis School faculty in 2017, she brought the African turquoise killifish along with her. With a lifespan of 3 to 6 months, this short-lived vertebrate has emerged as an exciting model for aging research.
So the fish is actually very pretty, very colorful. And so in the 1960s, an expedition in the Gonaraju Park in Zimbabwe isolated them. And so at that point, they sort of became like one of the best kept secret in fish hobbyists. And they're kind of quirky. They're not easy to breed. They need to be put on dry medium and then back on in wet medium. So they have some of those quirks, right? So people were willing to try to tinker with all of this because they were so pretty. And so they were bred and bred and bred. In 2003, there's this lab in Italy, the lab of Alessandro Celerino, who became aware of that fish and its special sort of life history. And the reason they're so short-lived is because they live in those ephemeral ponds of water that are only there during the wet season. And so he became aware of their ecology and of the fact that there were this strain that had been sort of kept by hobbyists. And because they're so short-lived, he thought, okay, maybe those are going to be a great model for aging biology. The reason it's so powerful is because we can recapitulate most of what you expect from aging in that short amount of time, including cognitive decline, increased amount of cancers, decreased fertility, and all of that. And it's not that they're dying of like something random. They are dying of aging. When we look at all the signs, they have all the signs of aging, which means we can recapitulate an entire human lifespan in less than 6 months. And so that's very attractive to study aging, diseases of aging, as a model.
In order to uncover clues to aging processes, Benayoun had previously worked with colleagues at Stanford to map the fish's genome and develop techniques to manipulate genes, enabling a way to better understand each of their functions, meaning that studies like those done in mice could now be done in killifish as well. This process yielded an interesting discovery. Similar to the human genome, the killifish had a large number of transposable elements, DNA sequences that are sometimes called jumping genes, because they can move from one location on the genome to another.
You can think of those as ancient viruses that have colonized our genomes. And so in the human genome, it's about half of it are those, and people used to refer to it as junk DNA, but there's no such thing as junk DNA.
In fact, Benayoun says that transposons are responsible for developments like the evolution of the placenta in mammals, and possibly the bigger brain and upright stature that differentiate humans from chimpanzees.
So there's definitely good things about transposons, right? But at the same time, if they're left unchecked, they're going to go and copy themselves. And copying is not a problem in and of itself. The problem is that they paste themselves. And they tend to integrate, so this pasting is integration, in regions that are actively expressed, which means they're going to mess up the expression of genes that you need at a specific time. One of the problems is they can integrate themselves in genes that are important for cell health, including tumor suppressor genes. And so when they go everywhere, they're going to create that increasing mutational load, and they can actually drive tumorigenesis, right, by turning off the safeguards against uncontrolled cell proliferation. That's one thing. The other thing is they look exactly like viruses as far as our immune system is concerned. They used to be viruses. And so when our immune system sees a transposon being expressed, it actually activates and tries to kill it. Now, the problem is it comes from inside our cells. And so that's actually a hypothesis that I have, that maybe part of the problem with aging is we have sort of an autoimmune response against those viruses. And we know that sustained immune response, which is, this inflammatory response, is actually very bad for us. The immune response is good if it's fast, and as soon as there's nothing, left to fight, it goes back down. And the problem is if the transposons are here and are being transcribed and transcribed and transcribed, it never goes down. And the increased inflammation also creates sort of the conditions for a lot of diseases to happen, like, arthritis and, again, cancer and even neurodegeneration, this inflammatory thing. So the hypothesis in my lab is that it's not just that transposons are getting de-repressed as a bystander of aging and age-related disease. We think that them being de-repressed actually may be the root cause of a lot of those aging phenotypes, which is why we think it would be very interesting to try to use the killifish to see if we can either decrease the activity of those transposons, because they do increase with age, or to trying to find a way to make the immune system oblivious to them.
Understanding differences between male and female aging is another area of interest to Benayoun, and one she says has been understudied, in part because women's hormone fluctuations can complicate research.
Which means that actually a lot of the drug development and health work has been done strictly on men. I think the thing that everyone knows is that the life expectancy of women is always higher than the life expectancy of men by about 5 to 10 years, depending on the countries. But what's really interesting is that NIH has created that program, which is called the Intervention Testing Program, where they wanted to create a standardized and robust way of testing drugs and interventions to see the impact on aging. And because this was really well done, they have 3 independent sites and they look at females and males. I think one of the biggest surprise and results coming out of the ITP has been that there's pretty much no intervention that works the same in both sexes. Either they work much better in one sex than the other, and that suggests that there's a fundamental difference in the way we age. Basically, when I first started, I was thinking it was majorly important that we look at both sexes.
This has yielded some surprising findings.
And I was expecting differences, but I was expecting that things that would be male-specific and female-specific would be of the same magnitude. So when we started, we were interested in macrophages, which are one of those immune cell types that are important for inflammation during aging. And we decided to map their transcriptome using our RNA-seq. And so we did that, and then we got the RNA-seq results back, and there was 20 times more things that were female-specific with age than things that were male-specific with age. And I was like, this is not possible. This is really weird. Again, because you feel like someone should have seen this before. I was actually thinking, okay, there must be a problem in the experiment. Let's just redo it. And we did it again in the same cell type and saw the same thing. And we did it in 2 additional cell types and saw the same thing. At least in immune cell types, aging creates many more changes in female cells than in male cells. That could explain a lot of the differences in, immune diseases. Young women are much more subject to autoimmune diseases like lupus. It's a frequency of 9 to 1 for women to men, but that decreases after menopause, right? So again, there's a huge interplay between hormones and immunity, and in immune cell types, we see that aging creates many more changes in females than in males.
Benayoun is specifically interested in the role of hormones and menopause.
Menopause is a major event in life in that it has huge public health implications. Now, a lot of people used to tell me like, oh, why do we care about menopause? We don't want women to be pregnant when they're 70. But again, that's not all the ovary does. Yes, the ovary produces eggs, which can be used to be pregnant. But the ovary is also a major hormone production hub, which has health impact. Now, that I'm creating my own research program, I really wanted to combine these 2 ideas of, okay, we know there are fundamental differences in the way women and men are going to age, and some of that might be hormonal. And the idea that organismal aging is a problem, and so basically bringing back the idea that we need to take into account the fact that females and males are different. Most people actually just study males, and we know very little about differential mechanisms. And that not just with aging, it's across fields. People usually just study males, whether human or mouse, because it's simpler. And so that's something that I'm very passionate about in my lab. And I'm sure my students are a bit, at first actually, they were a bit like, oh, why do we have to keep track of mouse hormonal cycles? And then they notice, wow, it makes a huge difference. So, we're studying aspects of immunity, and we're actually saying that the immune response is vastly different depending on where those mice are in their hormonal cycle. And so, yes, the data looks noisy, but if you're actually stratifying it by the hormonal status, it's not more noisy, and it gives you more information.
Beyond her specific research findings, Benayoun is advancing the idea that there is value in studying what others may have viewed as junk or noise.
Maybe it's just I'm a glutton for punishment, and I see something complicated, and instead of being like, oh, let's try to simplify this, I'm like, oh, that sounds fun. Let's try to figure out why it's important.
That wraps up this lesson in Lifespan Health. Thanks to Professor Bérénice Benayoun for her time and expertise, and to all of you for choosing to listen. Join us next time for another lesson in Lifespan Health, and please subscribe to our podcast at lifespanhealth.usc.edu.